Okay, so now let's talk about the bacterial cell. There are probably going to be some structures that you've never heard about. Okay. First and foremost, there is a list of bacterial cell shapes. There's a table in the lecture notes with terms like cocci, bacilli, vibrio, spirochetes. You have to be able to match a description of the shape and the name. Will I ask you about, like, what kind of shape does Clostridium difficile have? No, I won't. You see what I'm saying? Like, examples, yeah, I can give you an example, but at the end of the day, you can't memorize all bacterial species, how they look, whether they're aerobic, anaerobic, fastidious, non-fastidious, stuff like that. No, please, just understand the concept, okay? So if I ask you, like, rod-shaped, well, that's bacillus, okay? So we got this. We're going to go back to what keeps cells in shape. But first, we need to start with the structures found in all bacterial cells. There are no exceptions from that rule. Pretty rare. So in all cells, you're going to find nucleoid. Okay? So this is your DNA of the cell. Remember, these are bacteria. They do not have intracellular membrane-bound organelles. Bacterial DNA is not partitioned, it's not surrounded by the membrane, it is in the cytoplasm, but it's not diffused, it's just concentrated in some place. Clear? So, cytoplasm, okay? I mean, I don't even know what to say about it, it fills up the cell, right? Ribosomes. All cells have ribosomes, which are responsible for protein synthesis. And cell membrane. So while we are on the cell membrane, I want to address a couple of things here. It's going to be the transport across the membrane. Again, reminder how this whole thing works. So transport across the membrane can be passive, which we also call diffusion. And the feature of the passive transport is that it does not require ATP. So basically your molecules are moving from the high concentration to low concentration. Does that make sense? Like this ball is rolling from high to low. So molecules move along the concentration gradient. And then you have active transport. In case of active transport, molecules are moving from low concentration to high concentration against the concentration gradient. And this process does require energy, does require ATP. Okay? Now, not only it requires ATP, it requires special proteins that we call pumps. Okay, now, what about passive transport? In passive transport, you have two further options. One option is for molecules to cross the membrane directly, just like David Copperfield crosses the walls. So we call this simple diffusion. And simple diffusion is reserved for hydrophobic molecules, okay? They can cross the membrane directly, things like alcohol, for instance. Now, facilitated diffusion is for hydrophilic molecules. Okay, but facilitated diffusion requires transmembrane proteins, such as channels or carriers. Are we clear? Now, I'm positive that all of you have heard about this concept before. And it sounds very fundamental. How is it relevant to what we're going to be talking about? Well, for an antibiotic to enter the bacterial cell, antibiotics are usually large molecules, well, fairly large, very often hydrophilic. In order for them to enter the cell, they gotta have channels or carriers to diffuse inside the cell. Make sense? If the cell manages to reduce the number of channels, it becomes antibiotic resistant. Another option for the cell to become antibiotic resistant is to pump the antibiotic out. It will require ATP though, but it would work. Does that make sense? Any questions here? Good? Okay, moving on. All right. So now we're going to chat about the structures that are found not, wait, English problem, sorry, that is found in many, but not all bacterial cells, okay? So basically, disclaimer, I'm not going to write it down, but from now on, everything that we're going to be talking about, many, but not all. So the first is ridiculously simple. Inclusions. Inclusions are non-membrane bound structures within the bacterial cell that store things, okay? From nutrients to, I don't know, whatever. Next structure, and I almost guarantee you, well, you may have heard about it, I don't know. Endospore. So the main function of endospore is the DNA protection, but let's work out how it protects. So imagine you have a vegetative cell. The term vegetative cell refers to the cell that actively divides and actively metabolizes chemicals. I mean, it lives, okay? It lives. Now, when this vegetative cell is exposed to harsh environment, and I know this is a very scientific term, harsh environment, in reality, desiccation or lack of nutrients or presence of toxic chemicals like antibiotics, so something doesn't go well, some cells, some bacteria can form an endospore. So what they do, they form multiple layers, protective layers of carbohydrates, glycoproteins, protoglycans, and proteins around DNA. They do not include, like, a little bit of cytoplasm, all the DNA covered in that really, really sturdy shell. Does that make sense? And then mother cell dies. Endospores are extremely resilient. They can survive for years. To my knowledge, the oldest endospore ever recovered was 70 million years old from a piece of amber, okay? If you will swipe the surfaces of those cabinets and cubbies in the dust, oh, you're gonna find a ton of endospores, mostly of Bacillus subtilis, okay? Now, when the environment becomes good, and I know that's even more scientific term, so when toxic chemicals are gone, when nutrients are present, when water is abundant, endospore can develop into a vegetative cell. Okay? Does that make sense? Usually, this process does not cause any troubles. What causes troubles are a couple of terms here. Students confuse too, so I'm telling you, don't confuse them. So the process of forming the endospore, this process here, is called sporulation. Okay? The process of getting out of endospore into a vegetative cell is called germination. You know, kind of like a plant seed germinates. Are we clear? Okay, one important statement. that I want to highlight. Endospore is not a reproductive structure. Spores of fungi are, endospores are not, okay? This is the escape pod that DNA leaves, you know, the dying cell in. Questions about the endospore? Okay, good. Because next we're going to talk about cell wall, a little bit about osmosis, a little bit about the cell shape. And the knowledge of the cell wall structure is absolutely critical for our future conversations, especially about antibiotics. You can clearly see that I should have had F for the art in the middle school. This is why I like to use many colors. So cell wall consists of the chains of glycan. Okay, so this is your glycan chain. Okay, makes sense? This is a carbohydrate. This is the polysaccharide, right? And there are elements, you don't need to know the names of these molecules. It's N-acetylmuramic acid and N-acetylglucosamine. The point is basically you go NAM, NAG, NAM, NAG, you just disperse them. So you make one chain, if you're a bacteria, and then you make another chain, and you set them one next to another, okay? Now, is this a reliable structure? Of course not. It's like making a raft out of multiple individual logs without having any bindings, okay? It's not gonna work. So you need to cross-link those glycan chains, and you do that by establishing what's called a peptide link between them. So we say that cell wall is composed of peptidoglycan. Does that make sense? So you have carbohydrates that form glycan chains. You have amino acids that form peptide links, and overall we say that the wall is made out of peptidoglycan. Okay, we good? Now, why do we pay so much attention to that? Because the cell wall structure for bacteria is unique, and if we can target the formation of the cell wall and kind of disrupt it, bacteria becomes kind of naked and can die, and a good chunk of antibiotics that we use in clinical practice do exactly that. Penicillins, vancomycin, bacitracin, cephalosporins, carbapenems, they all target the cell wall formation. So it's going to be unit 3, don't worry about it now. I'm trying to highlight why it's important to know what we're talking about here. Turns out that cell wall in different bacteria can be different. And based on the structure of the cell wall, bacteria can be divided into 2 large categories, which are, I'm going to use fancy colors again, gram-positive and gram-negative. So these names have nothing to do with the metric unit of mass. They named after the Austrian? Well, some German-speaking fella, Hans Christian Gram, who developed a gram stain. And this gram stain allows us to distinguish those 2 big groups of bacteria based on their cell wall structure. If stain comes out blue, bacterium is gram-positive. If stain comes out pink, bacterium is gram-negative. Okay? Now, what's the difference? So if you would look at the structure of gram-positive, let's call it cell envelope, at the very bottom, of course, you're going to have cell membrane, okay? Gram-negatives, the same, okay? But then, in gram-positives, you're going to have a pretty substantial cell wall, okay? In gram-negatives, cell wall is much smaller. But gram-negatives have a third layer, which is called an outer membrane layer. Does that make sense? So you see that there are two distinct structures, cell membrane and pretty humongous cell wall versus cell membrane, fairly thin cell wall, and the outer membrane on the outside. Okay, phospholipid, peptidoglycan, phospholipid again. We're all on the same page so far? We're good? Why it's important? Gram-positives and gram-negatives have different resistance to cleaning agents, have different resistance to different antibiotics. If you treat gram-positive infections, you're going to be using things like azithromycin or various penicillins. If you treat gram-negatives, you may focus more on tetracyclines, carbapenems maybe, chloramphenicol to some extent. Okay, does that make sense? So you will select antibiotics based on the structure. So far we good? Now, a couple of unique structures found in gram-positives and gram-negatives. So first one is here. Cell wall in gram-positives is penetrated by teichoic acid. Teichoic acid is uniquely found in gram-positives. It's only in gram-positives, okay? It's a virulence factor. So this is the molecule that increases the adhesion of cells to this adhesion of bacteria to the surface of the cells, okay? It also stimulates the immune response. So far we good? And what about here, the gram-negatives? Turns out gram-negatives have a fascinating molecule that also sits in the outer membrane, okay, not in a cell wall, in an outer membrane, which is called lipopolysaccharide, or LPS. So LPS, that is found exclusively in gram-negatives, is a very potent stimulation of inflammatory responses. And it causes non-specific symptoms, such as malaise, fever, headaches, joint and bone aches. Does that make sense? There's a term in clinical practice called gram-negative bacteremia. When a patient has gram-negative microorganisms in the blood, when they die, they will release lipopolysaccharide. See what I'm saying? And the patient with gram-negative bacteremia develop those symptoms that I just described. They're fairly non-specific. Whether I have blood-borne E. coli or blood-borne Serratia or blood-borne Salmonella, which are distinct genera, okay, I'm going to have basically the same symptoms over and over and over. Does that make sense to everybody? Okay. All right, a couple more things. First of all, there are microorganisms without the cell wall. So, this is a crappy marker, okay? Mycoplasma, so known as molecules, contains no cell wall. I do want you to know that. Question?
I can't see what you wrote after
Mollicutes. It's a general term for microorganisms that do not have cell wall. Okay, and then there are, I'm not trying to make it confusing, believe me, but then there is a group of bacteria called mycobacteria. You definitely know one of the diseases that is caused by mycobacteria. It's tuberculosis, Mycobacterium tuberculosis. So mycobacteria, look, they look like this. Of course they have cell membrane, right? And then they have cell wall, pretty substantial one. So they do have, look, they do have gram-positive structure right here, okay? Good. But turns out, on the outside of that gram-positive structure, they have a layer of so-called, I'm running out of colors, mycolic acid. Mycolic acid is the waxy substance, okay? We're good? And it protects mycobacteria from, you know, many antibacterial responses in the body. It allows it to survive in the macrophages, it protects it from hydrolysis in the lysosome in the acidic environment. So this mycolic acid makes mycobacteria especially resistant. Are we clear? So here's the little deal. So is it gram-positive bacteria? Yes. By all means of classification, it is gram-positive. But can we stain it using gram stain? Turns out, no. We have to use a separate stain. Does that make sense? So this is why we make sort of a footnote saying, okay, mycobacteria, formally gram-positive. In reality, we can stain it. But structure-wise, pretty similar. Questions so far? Okay, little review of what is osmosis and how osmosis allows cells to maintain its shape and how cells can survive in certain environments and stuff like that. So let's chat about osmosis. It's going to be very short and sweet, and then we're going to take a small break. Osmosis, movement of water from low solute concentration, right, to high solute concentration. So this can become a little confusing. Remember, we are not talking about water concentration, we're talking about solute concentration, okay? So you can think of it this way. Water always tries to dilute something. Clear? So like if something is more concentrated, water will rush there. So what is the consequence of this? The consequence of this is 3 states. So you have, and I'm going to quickly ask you. So you know, you have isotonic environment, so environment, right? Environment that surrounds the cell can be either isotonic or hypertonic or hypotonic, okay? So what does that mean? In isotonic environment, the concentration of solutes inside the cell and outside the cell is the same, right? Good? So let me ask you this. What happens to the cell in the isotonic environment? Does it change shape? Does the water go in? No, because the concentrations on both sides of the membrane are the same. Nothing really happens. So, nothing. When concentration, listen closely, of solutes outside of the cell is higher than inside of the cell, what's going to happen to the cell? Shrink. I know this is the main confusion, so think about it. Think, okay, I'm the cell, the concentration outside of me is really high, so the water will move from me, from inside of me, into the environment. I'm going to shrink, okay? Therefore, in hypotonic, the cell is going to, let's say, swell. Let's not kill it yet, okay? Let's say swell. So turns out, virtually all microbes, some exceptions, exist in hypotonic environments, okay? Which means they should swell and, as you suggested, burst. Obviously, they don't, huh?
The cell wall protects it.
Bingo. Cell wall bacteria are protected from bursting by the cell wall. What if we throw antibiotics on the bacteria that will damage the cell wall? What's going to happen to bacteria? They're going to go boom, right? That's how we kill them. Now remember I mentioned protozoa, unicellular eukaryotes, and I told you that they are, well, I don't remember if I told you this, but they do not have a cell wall. That's the rule. So if they don't have a cell wall, and they exist in the hypotonic environment, how the hell do they not explode? So basically, imagine you are a little cell, no cell wall, you're swimming in a hypotonic environment, water constantly rushes into you. Okay, what you gonna do? If you have a basement that is regularly flooded, what do you install in the basement? A pump. So this is what those cells do. Protozoa constantly pumping water out. This is why if you look at protozoa, they just moving around, eating and eating and eating. Because in order to pump, you gotta have a lot of ATP. Does that make sense? Now, one of my favorite questions. Are you familiar with the Dead Sea in Israel? Very high concentration of salt, right? Guess what? If you sample it, and we'll try to analyze for the presence of bacteria, there's going to be a shit ton of bacteria there. Well, probably archaea mostly, but still. So how the heck bacteria can survive in the environment with such high concentration of solvents? Anyone can provide an answer.
Because the cell wall's protection on the
Nope. They should shrink, right? But they don't. Now, did I ever say that those cells live in hypertonic environment? No, I didn't. I said that they live in the environment with a very high concentration of solutes. So what these cells have to do? They have to have the same concentration of solutes inside. So the environment for them is isotonic. These terms, hypotonic, hypertonic, isotonic, they aren't absolute terms. You see what I'm saying? They're relative. So if I say that I am taller than, I don't know, my son, doesn't mean that I'm the tallest person in the world. I'm just taller than him. Somebody else is taller than me. You see what? And I'm going to be smaller than those people. So same here. If the concentration of solutes inside the cell and outside the cell is the same, it's fine. It's isotonic environment. It doesn't matter that it's high. They can survive fine. Are we clear about it? Any questions? Okay. Let's do it.